Consciousness is defined as the state of being awake and aware of one's surroundings while receiving two different stimuli and personally deciding to be aware of one of them in particular. You are conscious right now. You're receiving multiple stimuli and are choosing to focus on my voice speaking to you. But that could change. As different surrounding stimuli are introduced, you can choose to shift your focus from a given stimuli to another. But you already know this. How many times have you been sitting in class listening to your professor speak, but transferred your focus and attention from the professor down to your phone? You can hear the professor speaking in the background, but you do not fully comprehend it.
You see, we are capable of freely shifting our focus and attention while we're in a conscious state. But how do we do it? What even is consciousness? Where is it? Is it our sense of self, or does it drive that sense of self? Can we control our consciousness, or does it drive us? What is able to affect consciousness?
One commonly used yet mysterious drug, anesthesia, is able to affect consciousness. Anesthesia is a collection of medications that dramatically affect consciousness and the process by which the brain interacts with the world. These drugs are able to disable the mechanisms that are necessary for consciousness. Administration of anesthesia results in the user falling unconscious in a mental state between our perception of sleep and wake. Thus, studies on anesthesia are extremely useful in studying which parts of the brain specifically function to produce the state of consciousness. Furthermore, it shows how these parts cease interaction while one is unconscious. Understanding what anesthesia is and how it works is key to uncovering the stigma surrounding consciousness. It can begin to explain how consciousness works in the human brain, help us better define consciousness, and further understand the evolution of consciousness. Most people know the general physical effects of anesthesia on the human body. Anesthesia makes you unaware of your surroundings through sensory inhibition and suppresses or slows down autonomic bodily functions, such as heart rate, breathing, and circulation. Most significantly, it places you in a temporary state of amnesia. It has been difficult to find a unifying theory on how anesthesia functions due to the general mystery encompassing consciousness and in just the brain itself. There are obvious issues involved in visualizing and isolating each neurological component. However, through anesthesia research and EEG scans, we have begun to uncover specific components of the brain that work together to produce the state of consciousness.
Scientists have struggled to find methods to study brain activity for just as long as the creation of the neurology research field. However, scientists have created one of the most advanced pieces of technology available today, the electroencephalogram, or EEG. The EEG is designed to amplify natural brainwave emissions and record them onto a computer. To obtain the recordings, a cap with an array of different size electrodes is secured to the head. Before placing the cap on the head, the electrodes are soaked in a gel solution that helps amplify the brainwaves so that they can be picked up and measured by the multitude of sensors. The number of brainwave amplitudes are shown in many different areas of the brain and demonstrate how active the area is. The EEG does not measure individual action potentials. However, it can identify regions of the brain experiencing many action potentials, and therefore brain activity. There are two general ways in which EEG observations of anesthetized patients can be used to better understand consciousness. The first and most common observation is to put patients under anesthesia while using EEGs to study what areas of the brain subsequently shut off as a patient becomes unconscious. In such studies, researchers use EEGs to measure consciousness through brainwave patterns produced. Researchers then examine where the brain activity lessened or ceased during anesthesia administration. They can further explore these studies by titrating anesthetics into sub-anesthetic doses. Titration allows for a less severe but more precise experimental effect, which can be used to observe select aspects of consciousness such as memory, attention, pain processing, and emotion. Researchers have now discovered the underlying functions behind anesthesia. It suppresses activity in the thalamus and isolates it from the rest of the brain. In the human brain, the thalamus has a multitude of functional roles, such as being the relay station between other brain areas and all sensory stimuli, besides olfaction. Since anesthesia makes you unconscious through the reduction of thalamic activity, it seems likely that consciousness comes from the thalamus. Rather, it seems likely that consciousness is derived from the interactions between the thalamus and the rest of the brain. Regardless, it is clear that the thalamus is integral for the ability for consciousness, in a sense, exist. In the conscious mind, the state of alertness is thought to be due to signals that the thalamic region receives. After anesthesia is administered, the brain begins to feel tired and quickly slips into unconsciousness. In doing so, the thalamic region receives less signaling and emits a distinct oscillating brainwave pattern that denotes the start of unconsciousness. This oscillating pattern was found to occur between the thalamocortical and corticocortical areas. By observing EEGs in individuals falling into an unconscious state, scientists have discovered that consciousness is mediated at the level of thalamocortical interactions, particularly in what is considered the thalamocortical and corticocortical reverberant loops. This figure shows the interacting parts of the brain that may contribute to consciousness. These are also the parts that emit oscillating brainwave patterns during unconsciousness. Researchers have broken down the effects of unconscious-inducing anesthetics into 6 substages. First, sensory inputs in the ventral tegmental area, or VTA, result in depression of the ARAS, or the ascending reticular activating system. This causes a reduction in acetylcholine availability and allows for the release of GABAergic inhibitory responses. This results in the second step, in which the limbic system becomes less reactive and does not interact with the dorsolateral prefrontal cortex. This prevents the ability for recent memories to transfer for storage. In step 3, acetylcholine is further decreased, ultimately resulting in the closure of thalamic gates. This leads to step 4, in which reverberations in the corticocortical and thalamocortical loop are blocked. This results in the uncoupling of parietal prefrontal cortical interactions. And lastly, the uncoupling of parietal prefrontal cortical interactions causes depression of the prefrontal cortex and a complete loss of consciousness. Studies focusing on the inactivation of the conscious area of the brain through use of anesthesia have allowed researchers to conclude that in the human brain, the main source of consciousness is the thalamocortical reverberant network loop.
The second, less used way EEG utilization allows for research on consciousness is observing patients regaining consciousness after anesthesia by studying the order of which brain areas reconnect after coming out of unconsciousness. It is assumed that the parts of the brain that reconnect first are the primitive aspects of consciousness. Information on emergence from unconsciousness in humans can be used to understand the evolution of consciousness. It has been previously noted that EEGs may be better able to measure the differences in a conscious and unconscious mind as a patient comes out of anesthesia. This is due to the fact that people typically regain their state of consciousness after anesthesia-induced unconsciousness quicker than the time it takes for anesthesia to induce unconsciousness. Many researchers argue that studying how consciousness is regained is superior to studying the transition into unconsciousness, because anesthesia may cause nonspecific deactivations of the brain that do not necessarily play an active role in consciousness. Through EEG research on both non-human animals and humans, subsequent data have characterized the awake state as a pattern of desynchronized high-frequency and low-amplitude activity. Researchers then recorded EEG data following the use of anesthetics. The subsequent data suggests that the unconscious brain pattern is one of low frequency and high amplitude activity. Thus, scientists summarize that neuronal activity in the neocortex is particularly required for consciousness. Consciousness appears to be linked specifically with neural activity in the thalamocortical system. Additionally, the midline brain structures in the brainstem and midbrain are essential for keeping the cortex in an aroused or awake state, and thus the cortical regions serve as specific contributors to contents of the conscious experience.
Mirroring the previously mentioned studies, these researchers found that the thalamocortical oscillations are critically important for consciousness. These oscillating patterns help integrate cognitive modules in the brain that are spatially distant and functionally diverse. The long-range connections that are assisted by the thalamocortical oscillations are mainly important for the experimental aspects of consciousness. Simply put, they are critical for not only being awake, but for the ability of awareness, as well as creating memories. These are the areas that are also suppressed while under general anesthesia. Researchers note that when consciousness emerges, most brain activity is in the brainstem, hypothalamus, thalamus, and anterior cingulate, particularly frontal parietal connectivity. In the same study, researchers assessed their findings in humans in terms of evolution. They used anesthesia as a tool to turn back the evolutionary clock of cognitive function in humans. They used the tactic of altering anesthetic exposure length to alter depth of unconsciousness in order to observe the return of neural function to model phylogeny. In their studies, researchers found that phylogenetically ancient structures in the brainstem limited neocortical involvement, yet were still suffice for primitive consciousness. Researchers essentially reverse-engineered consciousness to postulate that early mammals and birds possessing the structures that first function in human consciousness are also capable of consciousness itself.
Researchers have found that the mesial parietal cortex, posterior cingulate gyrus, and precuneus gyrus are more inactive in individuals under anesthesia-induced unconsciousness. These areas are often the first areas of the brain to recover as anesthesia wears off, and they may be more evolutionary ancient precursors to consciousness.
Based on current studies on the topic, it can be concluded that consciousness arises from the thalamus and thalamocortical and corticocortical oscillations in the brain. It has been found that vegetative patients and patients under anesthesia experience the same thalamocortical and corticocortical disconnection, which is evidence that unconsciousness that arises naturally and anesthesia-induced consciousness are similar, and anesthesia is a valid and helpful tool for learning about consciousness. Although research of this type is uncommon, the results are compelling. With the help of anesthesia, researchers have not only found the mechanisms involved in human consciousness, but that these mechanisms are also found at the earlier points of vertebrate brain evolution. Furthermore, consciousness, or the ability to experience the world, differs among animals in its degree rather than in the type. As of now, research on anesthesia-induced consciousness has been limited to non-invasive studies. It is too dangerous and certainly non-popular to invasively study the brain and subsequent results from anesthetics. It has been suggested that EEGs do not show all the details necessary to understand what specific areas of the brain function to create consciousness. Although we may know the parts of the brain responsible for consciousness, if there are specific sections within these parts that are the truly underlying primary functions of consciousness, we would not be aware. EEG scans are still too weak to truly measure subtle differences that could be the main differences between the conscious and unconscious mind. We suggest that it may be beneficial for researchers to also use in vivo calcium signaling to observe the function of specific cells in these areas of the brain. It is hard to quantify something we don't fully understand like consciousness, but as technology develops, we will be able to conduct more targeted research. In doing so, it will become easier to understand what implications anesthesia has on the conscious mind.